I'm Susan. I'm happy to be here. I'm really excited to follow up on the speakers I'm just following up with. That is spectacular and a beautiful setup for everything I'm going to tell you about. I have two parts and I talk fast, but I promise you it will be worth it. Please do interrupt me, but maybe in the second part more than the first. So I'm going to start with talking about a perspective Julie Terrio and I wrote a couple of years ago. And that is going to be a bit of philosophy, so that's really great for coffee. And then I want to tell you about an actual data-driven example based on those themes, and I really want to get to that, and that's the second part. So I shortened everything. Okay, thank you to the cell science team, especially the authors of what we affectionately call the Endo paper. This paper is going to be hopefully up in about a week. I also want to thank our collaborators, Julie Terrio and Jin Wang, both of which are in the audience today, which doesn't happen very often, so this is extra exciting. So I'm going to take a very cell-centric point of view and focus on the cell as the basic unit of life, because nothing smaller than a cell is alive and nothing larger is any more alive. The cell is an ideal level of biological organization to integrate all types of observables. So if this works, there we go. So what do we need to know the cell or understand the cell? Not just predict the cell. Let's assume that we consider all of the cells in one particular organism, say a human, and we're going to therefore fix the genotype and focus on just the phenotype and the environment. And for this talk, I'm going to redefine now from the cellular perspective, the cellular phenotype, which are all of the observables that we can see and measure that are intrinsic to the cell, and the environment is everything that is then extrinsic to the cell. So I'm going to define cell observable for you. It is anything we can observe about a cell, and it sits in between what is actually happening in the cell, which we may not have access to, as well as what we can actually measure and then work with. And that's something philosophical for us to come back to and has been alluded to actually just by Jianhua a moment ago as well.
Susan, can you at least tell me what am I looking at?
Oh, this is just a picture of a cell. This is a realistic rendition of a stem cell. It's just there.
What are these organelles?
All the organelles.
Okay.
Yes. We can talk about that at coffee too. So if we imagine all of the cells in a body, they can all exhibit many different cellular phenotypes, which we have for convenience sake classified as cell types such as epithelial cells or muscle cells, and then further into cell states such as proliferative, motile, quiescent, etc. And here I'm going to do, here we go. But defining cell states, of course, is very challenging, in part because of the heterogeneity among all of the individual cells in a population, which we've been hearing about a lot, and the continuous nature of the cell state landscape. So I skipped like 10 slides now to get to this punchline here, which is to say that we really want and believe that now is the time to update the concept of cell state to be a holistic cell state in which we bring together the quantitative spatiotemporal observables of the cell with the molecular observables and use the same formalisms that have been used a lot in this molecular space. And so traditionally cell states have been defined by what cells look like, which we're going to call cell organization, where cells are located in the body or in a tissue, which we're going to call environment, what cells do. I should give you guys the definitions while I go here, which is cell function. And then importantly, incredibly usefully for us over the recent past, is the molecular census, or all the molecules and how many there are in the cell. And we've used that to create signatures and representations of what we then call cell states. As Julie showed you earlier, we believe, and I think many of you do here as well, that if we understood the bidirectional interplay between, say, just molecular observables, but more broadly, holistic cell observables and their behavior in time, that we would start to get to understanding a cell.
Can I ask a question?
Yes.
So immediately my guess at the number of cell states is infinity, if like they're defined in terms of anything that I can observe.
We will get to that. That will be exactly where we're heading. I would say no. That is if you define it mathematically with a cell state variable, but I would say that that is not necessarily the case.
For example, some things that I can observe seem not phenotypy and part of the genotype to phenotype map, right?
So which measurements we use to understand what the cell is doing, that becomes the question. We'll get there. Let's go through the example and come back to that.
Okay.
So a really quick example, just to say what we mean by something that is measurable and observable, is we take the molecular census, which we are all most commonly used to talking about in a measurable way. We have the transcriptome, the proteome, lipidome, et cetera, and we have dynamic changes in the census. We can't always measure those dynamic changes. That's harder to do, but we certainly know that they're an observable that we would care about. What we can measure, for example, is, say, the proteome. We have individual proteins. We can imagine measuring for every cell, every single protein, how many there are. We can do this in these other categories of observables as well, where we can imagine, for example, the location of all of the different proteins, but also into these structures and assemblies such as organelles that have functional purposes, all the way to the folding of the genome and the morphology of the cell. And for those, of course, we can observe many things. We can measure a subset of those. For example, we could measure the total Golgi volume per cell as far as the mean and the standard deviation in a population or two populations of cells. Yes.
But I guess when you go back and forth between cell organization and your census, right?
Aha, wait 2 seconds. Okay, a note about categories. Any discretization of a continuum is imperfect, and if you are spending your time trying to decide whether something belongs to molecular census or to cell organization or both, don't worry about it. This is intended for us to be able to have conversation. It is also linked a lot to the types of data modalities that we use, which tend to be distinct. It is just a formalism. Is that what you were getting at?
I was, but maybe the point. I guess it's more a point to the question. But you know, for the census, for the sensi we make, right? We have unified profiling technologies, right? We're generally going to use Illumina sequencing, or we're going to use mass spec from Thermo, right? If you start looking at cell organization, right? Let's use Golgi as an example, right? Whether I do fib milling and image the Golgi, or whether I'm doing light microscopy, right? That's going to be completely different, right? So how do you?
Aha, yes. So this is exactly, normally when I give a talk, I have a slide that talks about the fact that we know how to represent the molecular aspects of the cell with our data. We don't have the light representations necessarily for cell organizational observables. And the point that I would make there is to say we need to create those if we want to understand the cell that way. So thank you for that. That would be a slide that I would have in a longer version of my talk. So if we now take all of these categories of observables and we now actually do something with them that you guys would be more used to, which is that we're going to say we're going to have them, we're going to have a vector of observables, and they're just assigned to categories loosely. And then, of course, we start thinking about all of the bidirectional feedback between these. We start having what we're used to looking at in systems biology and regulatory networks and in neural dynamics. The difference is that we're saying, just like was pointed out, that we want to have measurements that take care of observables that exist in these spaces that are not just molecular.
Maybe I want to ask the same question again, which is, you know, I mean, it's from just a philosophical point of view, molecular census is defined because you have molecules that come in numbers. What is discrete states of cell organization? It's a continuous you know, these are not discrete objects, they're continuous objects.
So let's take the data example that we have and then come back to the philosophy based on us trying to actually show you what we would do with observables that are coming from organization and behavior instead of from molecular to define or demonstrate whether we can actually define cell states in the standard sort of approach that people use of dynamical systems that you guys all used to. So can we come back to that at the end? Great.
And maybe you'll get to this too, but the way that my very simple brain has to think about this is there has to be a hierarchy of how we decide what's inclusive in your interesting set of parameters you're measuring about a cell. And we pull lots of things through cell function because that's how you do it in the genetic space a little bit, right? There's a lot of variation in a cancer genome, for example, but not all of it's functional. It doesn't really matter, right? It's all a passenger. Are there ways that you guys think about, maybe you'll get to this, that you can hierarchically organize?
You guys are so great because you have the setup for every single thing that I'm going to say next. So I'm going to say it maybe not in exactly the words you need. It is the basis of this data-driven framework that we believe that there is a set of observables and measurements that we will be able to get to represent the underlying living system that we can do, for example, from the lens of the molecules, and where we need many, many molecules to measure. For some cell states, they may be able to be described in a much simpler way with other observables that some of the molecules are signatures for, as opposed to actually defining. That is an open question. And in that particular research question, we need to start playing with and testing which types of measurements do we select in order to ask and define these states. And this is where the underlying sort of true state variable versus measured observables that we select in order to create a particular state space to do a particular experiment to ask these questions depend on each other. So it's absolutely valid to say we can come at it from a functional point of view or from a molecular point of view or from an organizational point of view. It is the question of how these interact with each other that we want to propose we pay attention to see what we can learn. And I'm going to give you an example of that. And before I do, I'm just going to say this is exactly why we need quantitative observables in these other categories. So we can start to identify what the best representations are that actually then represent and predict and help us explain cells. And as has been mentioned by John White just a moment ago, we will never have a complete set of observables. So we do need to then figure out, and there are ways to do that, to work with sparse data and to ask those questions. But to do that, we also need to have states that we're going to actually look at. And so we're going to get there. Our proof of concept that I'm going to move into now is towards building a dynamical holistic cell state landscape using dynamical systems framework that many of you are used to, because that theory supports non-equilibrium nature of cells. It was developed because of mechanistic understanding of gene regulatory networks. It's been applied primarily to molecular census data, but has also been applied to morphological data, for example, in EMT with Zhenhua and Eva Palush have done and others. And we, in this next section, want to apply this framework for cell states to cellular observables extracted via AI by time-lapse microscopy data. And so this is a particular data modality we're choosing because it gives us access to measurables of cell morphology, organization, and behavior, so that these are all accessible. It allows us to have direct measurements of the velocities needed to create the data-driven dynamical system. And what we then need, if we want to try to ask this question in the absence of focusing on the molecular census changes, is we need a model system with a clear visual phenotypic cell state change, not cell type change, and ideally one where the state transition occurs along a controllable axis independent of time. And so I'm going to introduce you guys to that model system and to what we're going to do to ask the question whether with these completely non-molecular image-based observables, we can use this type of framework to show what these cell states do. So I want to just acknowledge our four co-first authors, including Aaron Giolini, who has actually been mentioned already by Sui, as well as my long-term scientific collaborator, Matthias Liana, now 20 years, going on 20 years together. Okay, what is a model system? They are hiPS-Derived endothelial cells. Endothelial cells usually line blood vessels, and so they experience a lot of shear stress, and they behave and respond to the shear stress. We're going to image these in byte field, and we're going to image these by tagging with GFP the protein, the adherent, that forms the adherence junctions, the cell-to-cell junctions between the cells. When we put these cells into a flow chamber, and you can see in red, actually, I don't know if I have a pointer. Whatever, you can see the little red box on that flow chamber, the area that we image. These cells respond to the shear stress here. This is low shear stress at 6 dyne. They start to align and move upstream towards that flow. But we can now define 2 states that we predefined that we're going to ask questions about. So it's important, as I think a previous speaker mentioned, we need to know where we're starting and where we're going in order to do these experiments in a rigorous way and ask these questions. So here's the low shear stress. The morphology of the cell is that it's elongated, it's oriented parallel to the flow. The behavior is that it's migrating coherently upstream. And then one aspect of organization that we see is that you can see these blobs that are at the trailing edge of the cells in the retraction fibers. When we change the shear stress to a high shear stress, we get a completely different phenotypic cell state, as we can see in the microscope, in which the morphology is now still elongated, but oriented perpendicular. The cells move in an incoherent manner, and the blobs are no longer persistent. We are going to characterize and determine whether these states can be, these conditions can be used to identify stable cell states in this sort of formal way, and then ask the question of what happens in the middle. And since we may not get there, since everything here is running over time, we have 2 possibilities. You could imagine that there would be a saddle node bifurcation as you switch from one state to the other as you change your stress, so you're looking at the change in the vector field itself, or you could imagine a continuous transition. And spoiler alert, this is a data generated from our data and shows that there's a lesion of bistability at intermediate shear stress magnitudes that is consistent with a bifurcation of phenotypic cell states. And if we will see how far I get, I will describe to you how we get those components that are on those axes and how we create that vector field. So we started, of course, by trying to do this in a classic way, where we want to try to get the features, track and segment the cells. This becomes really tedious, even when we project these cells into 2D, which we can do because even though we image them in 3D, most of the information contained can be taken into account in 2D. So we said, well, can we actually learn the features that are important instead of pre-imposing exactly how we're going to measure these aspects of the cell state that I've already defined? To do that, we start with our Z-stack time-lapse movies. We're going to now just take snapshots to train the model. We're going to do a projection. I'm going to play the movie here. So we take 3D byte field and we do a standard deviation projection, which is incredibly powerful for pulling out a lot of the features that actually exist in the byte field, Z-stacks. And we're going to do a normal maximum intensity Z projection to get 2D images of the V adherent. Then we're going to use small random patches so we can commute on the cells, and the size is selected so that they contain one cell and the environment around the cell. And then we're going to use a diffusion autoencoder architecture in which we have a semantic encoder that conditions the image, denoising very close attention, and that we're going to do just on the byte field. Okay, and for that we're going to get a compact representation of the bright field co-ops. We're going to use that to condition the ability to make a noisy V-coherent image, have the prediction of the noise, and then the reconstruction of the image. And so by trying to do that, to go from the noisy V-coherent back all the way to the denoise V-coherent, we get the information that's contained in the bright field that actually allows us to do that. And that means that we have an interpretable readout for vector embeddings because this is a generative model and we can actually look and see what we see in the latent vector. Once we have trained the model, we only need the bright field to get these representations, which becomes very powerful in a generalizable way. That wouldn't be the case if we were focused on the V-coherent itself. So now we take all of our data sets that we use for training all of the images. We make these random patches, we run them through, we get a latent space. We are going to do PCA as a first step. We had a 512-dimensional latent space, and we get a feature space. When we look at these PCs, we can see that actually the top 10 PCs only on average cover about 2% each. We only get to about 95% as variance with 97 PCs. And we can start looking at a lot of these measurements that we didn't do in the standard way to start understanding which of the PCs might matter. We find that only the top 3 to 4 PCs seem to capture the biologically relevant variation for a slew of the measurements that we expect based on the actual observed phenotype that we see.
So
We
what are
can-
the other PCs?
What?
What are the other PCs capturing?
Here.
Oh,
We can take a latent walk, reconstruct the V-coherent co-ops along the PC axes. And when we do that, we can visually see that only those first 3 PCs start to show us things that we might interpret as biologists as mattering for this phenotype. And after that, it's not clear exactly what we see. And actually, Yogesh, we can come back to that because that would be a different setup for the model to really get at that. Yeah.
Do you see any spatial correlation
Yeah.
along any of your PCs?
So let's come back to what happens spatially in a moment because you will see, yeah, let's come back to that. There are different ways that we could set up this model, which we have not done, that would also allow us to ask questions about which spatial scale matters, et cetera. But that's a great question.
Is this dynamic data?
Yes, it is.
Have you tried dynamic mode decomposition? Things like
We have not created a model based on the dynamics.
No, dynamic mode decomposition is like kind of like
No, we have not, but we need to. Okay. Can we disentangle the representation of these two, where you can see that there is an entanglement of how elongated the cells are versus how oriented the cells are? And we do that just with a polar transformation. And then we also are going to define theta and R, which theta is sort of representative orientation, R is representative elongation, but these are now ML derived, and these are at the patch level, so these are not standard measurements that you use normally. And then rho, we're going to define as negative PC3. And when we do that, we have a beautiful latent walk along all sorts of aspects that we visually know matter for these two states, so we can start to create a morphological cell state space that then we apply the dynamical systems framework to. I want to point out that here we have used morphological features based on training a model on snapshots. Even though we have temporal data, we can apply this in time now. So how do we do that? We're going to have another challenge, which is that we don't want to and can't track all of the cells. But we're going to actually create our trajectories in a grid-based manner because we've chosen the patch size to capture one cell within its immediate surroundings. And because over one time point to the next time point, we move a very, very small distance. We have done an extensive control where we compare this to cell-centered patches. Please talk to me about that at the end. This is incredibly important for us to be able to make biological interpretations out of this approach. We can then compute the displacements of the morphological state vector from one time point to the next. And then we can compute the values of the data-driven vector field as a conditional expectation over morphological state space, which we do in a way where we use a kernel-based method so we can smooth and get a reasonable computable space. If we look now in our 3-dimensional morphological space, we can look at 6-dyne and 21-dyne, and I can give you an example. We do identify one stable fixed point in all replicates in each of these conditions. And when we reconstruct where we are in that space, we get what we would expect based on what we know about the biology. So this is great. One aspect of our observed phenotypic cell state is accounted for. But as I told you, we also have this migration coherence. Spatially, we have a lot of coherence. This is coming back to that. And we also have the V-coherent blobs. So what do we do about those? Here's an example of coherent migration. Why is it not playing? Okay. And of uncoordinated or incoherent migration. And we can use optical flow between consecutive time points. And then we can normalize the vectors and we can get a score, a migration coherence score, where very high migration coherence is 1. And fortuitously, after lots and lots of looking at our data, we also found that the blobs, the persistent blobs, are incredibly closely associated with migration coherence. And so we're going to use that as a proxy for those as well, which you can see here if you look at the larger sort of image. Now we can do this on every single data replicate that we take. Here you see 3 for low and 2 for high. I'm going to show you the intermediates in a moment. And so you can see that parallel orientation, blob persistence, and high migration coherence versus low on the other side coupled together. We can see that all the cells are sort of elongated and that the density is mostly an experimental source of variation. I already went through the slides, so I'm going to move forward. We're now going to ask how might the dynamics change as we change the axis of shear stress and each point allow the system to relax back to steady state. And when we do that, we actually get, for 9-dyne, as we start moving in, we're going to get something that looks just like low shear stress. When we go to 12 and 15-dyne, and in each case, remember, we can tune the shear stress and we can get the dynamics in time. We get a mixed response where some replicates behave as you would expect, and some replicates start to exhibit a very mixed phenotype. This now gets to really interesting spatial questions, which we can discuss at the end, where the morphology is now elongated and locally oriented in different directions. The migration is all over the place, depending on which replicate and when you are. And the V-coherent blobs can sometimes be there and sometimes not.
So then can I ask you a few questions? So how, I should have asked this before, but how do you change your shear stress? Is it by changing the flow direction or something?
No, just the magnitude of the shear stress by changing the magnitude of the flow over the cells.
So how long does it take if you change in the middle of?
Yeah, so we have not dealt, those are exactly the experiments we want to do eventually. For this paper at this time, we start from no flow and go to each of the flows. We do not go from one flow to the other flow. That is something that's going to become very interesting in the future.
Do you have a hunch of whether the longer they're exposed to one type of flow, they're going to take a hot second?
Yogesh, I love your questions. All sorts of questions about memory in the system, about whether you're secreting ECM that creates, I mean, all of this comes up as you go forward. Yes. So you are already on the next paper.
But also there's the question if one is more stable. One of these flows is more stable. So if you perturb it enough times, you're always going to go to the most stable solution.
Yeah, I mean, we do not have the answer to that because we have not done that. What we have done is we have taken cells at low flow and moved them to high flow. And we've taken cells at high flow and moved them to low flow. Both of them respond in the way you expect. They respond similarly in the way you expect. However, what you're actually doing there is you're taking them from the low flow landscape, low flow vector field and doing a jump right to the high. It's not the same as if you actually did the tuning down to ask those questions. And that is a difficult experiment to do that we will do in the future.
You know, some of these things we also, so we have a project in aorta and we see the same thing. So in areas of high shear flow, you get a lot of exactly on the right side, what you see. And in the laminar flow region, you get it in the direction of flow.
Yeah, so this is a known thing, you know, in blood vessels that also when you have this higher shear stress that comes, you know, functionally that might be a response that's like to help sort of the rigor of the vessels themselves. These cells are baby endothelial cells, and whether they're purely 100% endothelial is not clear. They respond to much lower magnitudes of shear stress. Let me go 2 more slides and then come back to that because that gets really interesting. So this is, in 2 seconds, I'll get to my very favorite slide. Here's a 3D vector field at one particular replicate that actually shows 2 stable fixed points, one which looks very parallel and one which is actually stably at diagonal orientation for the entire replicate. And now we can actually look at many of the replicates and we can start looking at what happens to them. So you can see that as you go from either high or low shear stress, you have parallel orientation high migration coherence versus perpendicular orientation and low migration coherence into a bistable regime. And you can also see that as you're in these intermediates, you can start really decoupling some of these observations, where here you have parallel orientation and low migration coherence versus perpendicular orientation and high migration coherence. And if you look at the degree of elongation, we can in these regimes now see cells that are much less elongated, but some still have really high coherent migration, which gets very mysterious. What that all sums to, which is what makes this so exciting as we move forward, is that the association between these observables changes. So we can start getting at sort of the mechanism of how these interact with each other during the state transition in a really stable way, because we can actually pin them at each of these shear stress, or because we can do those experiments eventually where we can tie them, pull them in. So you already saw the spoiler and the conclusion. And so I'm just going to leave up a set of future directions, many of which have been touched on. And I think that where we're going to go with this in the context of how this compares to molecular observables is if we're asking questions about cell state, that we want to be able to maybe extract from data such as image data or dynamic image data, we can actually get pretty far and we can use the same formalisms. Oh, and I was right on time too. Okay, wonderful. So I want to thank you all for your attention and take questions or not for coffee. Yeah, go ahead.
I guess I'll ask the philosophical question again, because
Yeah, let's do
I
it.
think the soft, let me channel my best soft matter physicist, active matter, soft matter physicist. I would say what I would do is I would make a 3 order parameters. I don't know what background you come from, but you know, we know there should be some fields that change. My 3 order parameters would be exactly the order parameters you made. And then, in fact, I would posit some low-level hydrodynamic equation, and what it would tell me is that it would help me because I would have symmetry that would help me in the shear flows. And as far as I can gather, this is a very maybe more generalizable, elaborate way, but what you've reproduced is basically what if I gave Lisa Manning this thing and said, think about it for 10 minutes, this is what she would say to do. Or if I gave it to Christina, it doesn't matter. I can give you a list of people who are not me, who would be able to do this. Vincenzo down the road, right?
Yes.
So I don't understand, because I feel like, you know, it reminds me of the Sidney Brenner quote, and I'll be, you know, which is like, it seems like you're saying we should just do more, but I think less is best, right? Is this famous thing he says about Turing. So I feel like you're doing very elaborate soft matter physics to me, and I would like to bring it back to the kind of questions everyone else was asking. Again, it's just my impression from my background, so I would like to understand better.
No, that's great. That's wonderful. I come from a cell biology background. I certainly don't come from creating vector fields from images. I think one of the things that we're trying to achieve is the fact that we ended up using this approach at what we would have created from the other direction is actually what we find interesting here. We're trying to test whether we can use frameworks where we're going to now just look at the images and now take cell states where we couldn't necessarily completely define in advance what we might try out in multiple different ways and get at that answer. So Ihsan Nazogdast, who's our explanatory modeling director at the Institute, and I and Julie Theriault and others have lots and lots of conversations about all the cool sort of physics forward ways that we could analyze this project. We've talked with Margaret Gardell and Vincenzo about, you know, all of the exciting things that comes out of this. But as we were going, we didn't know much of this. We actually had the model telling us and proposing to us where we might want to head with this. We didn't even know what our phenotype was. We were asking the question, if we take this formal approach and ask, can we identify cell states in this way, making these definitions, would we get a cell state at all, a single fixed cell state, and what would happen in the middle? We could approach this from the biological side about asking questions about the underlying mechanisms in these cells in a different way, 100%. And it's actually the coming together of these that we're trying to use to establish ways to get at the complexity that comes in the data in our images while maintaining a biology or a physicist in the loop to try to do that. I do want to ask Julie, do you want to add anything since this is near and dear to your heart as well?
That was great. I think there's been a difficulty in the sort of quantitative image analysis space, I would say, for, you know, the past 15, 20 years, where a lot of people come up with nice computational methods for pulling out what we call image features that are things that basically cell biologists would say are important about a cell.
What cell biologists would say are what?
Are important about a cell. So things like the size and shape of the nucleus, you know, the orientation of the actin, things like that. The problem with that is that's still a curated list that's based on human prejudice. Sorry, that's still a curated list based on human prejudice of what we think is important and what our eyes are drawn to, largely because of our macroscopic training about understanding, you know, different objects in our visual fields. And so what we're aiming for here was to take advantage of things like the diffusion autoencoder so that you can use a computer vision based way that didn't walk in the door with all of the assumptions and the prejudices that humans have and ask the question of whether or not it would converge on the same thing that human cell biologists would converge on. And in this particular case, it did. And we actually find that encouraging because it means, you know, we're doing the math right. And also that the things that a human would say, looking down the microscope, oh, look at that, that's different, are also the same things the computer would say are the most different. And as Susan showed you, you know, that doesn't go down very far down. As soon as we start getting into higher dimensions, that latent space, that all goes away. So, you know, we've had a lot of discussion so far today circling around, well, what is the dimensionality? What is the intrinsic dimensionality of these systems? And I think it's useful, as came up in the gene expression space, to distinguish between dimensionality of stuff that we can measure, so 20,000 dimensions in genes, versus, dimensionality of things that actually give meaningful biological variation. And so in this case, absolutely, we converged on the things that we were expecting to converge on. But I think that's a positive result, because that gives us hope that then as we start getting into other kinds of spaces where we don't have such strongly informed, pre-existing assumptions about what the nature of the variation is, that we might still be able to rely on the computational approaches if we can convince ourselves from examples like this that they're robust.
Yeah, take away the opposite.
What do you take away?
Well, I just don't think there's a view from nowhere. I think the reason it works in this setting is precisely because humans could easily do it, because it's hydrodynamic, there's symmetries. There's no reason to think you can extrapolate to new settings where we would be lost. I don't see why computer vision or AI can do things in places we can't do stuff. I mean, at least I have not seen it. I view it as an automation tool or as an interpolation between two large database tools.
Super interesting question. I can't wait to have a fight about overclapping.
Sorry, Carson.
Oh, sorry, Carson. Do you want to? Sorry, I just want to make sure we have.
Okay, I put it out. Two questions. So first, to that point, if you don't take the first 3 or 4 PCs, but the other ones, and do the same thing, you also find 2 states. Second question.
If we've actually done that, that's a really good control. What I suspect is we will find something different. We will have different features for our axis if we train this model in time, because we will have temporal features to add in. And that's going to get really interesting. Okay. So right now we have those 3 PCs. Those are the 3 that are used to from snapshot. But the moment we add time in, it might get more, because now we're going to have features that might be sort of, locally changing. And that's where, so then we might actually need more PCs, and that's when it's going to get very interesting.
And if I made one last one, your axes are basically phenotypically, right? From that analysis,
I can't hear
you have phenotypic axes here, right? Can you replace that with molecular axes? I mean, can you do protein gene expression or whatever and do the same thing?
So in these cells, they are still the same type of endothelial cell, derived from the hiPS-Cs. They're not changing cell type. There may be signaling signatures we could use, and we need to look into that in the future.
Or couldn't you learn them? I think is what he's getting at.
Yeah, I mean, you would need something to learn with, right? So you would need to create a data set where you have that, and you would need to have a difference.
You would need to be able to see if there is one, if there is a map. Yeah.
Yes. Correct.
Yeah.
Cool. Thank you.
Thank you.